Hi, I am Sohail. Today I am going to talk about structure and composition of Golgi complex. Main points of the lesson are structure and composition of Golgi complex, functions of Golgi complex, and difference between Golgi complex and dictyosomes. Golgi complex modifies and packages proteins produced on rough endoplasmic reticulum. Golgi complex, they are present in eukaryotes like animal cell and plant cell. They are absent in prokaryotes like bacteria. Golgi complex, also known as Golgi body or Golgi apparatus. Golgi complex are called traffic director for cellular proteins, packaging and distribution center for specific destinations like lysosome formation. Golgi complex, it receives protein from endoplasmic reticulum. That protein is modified in Golgi complex, then it is packed into transport vesicles, and these transport vesicles are destined to different components. Look at this slide. Endoplasmic reticulum, they are present here in the cytoplasm. Here is rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum, they contain ribosomes, and ribosomes, they are involved in protein synthesis. That protein is packed in small vesicles which are called transport vesicles, and transport vesicles, they take that protein from endoplasmic reticulum to Golgi complex, where it is further processed and the finished product is packed in secretory vesicles. Golgi complex, their shape is like hollow dinner plates. Golgi complex, it consists of stacks of flattened membranous sacs called cisternae. Singular is called cisterna, and each cisterna has internal space that is called lumen. So, in the Golgi complex, there are many flat membranous structures are present which are stacked together, and each flat membranous structure that is called cisterna, plural is called cisternae. Inside each cisterna there is a cavity that is called lumen. Golgi complex, it has three areas, that is cis phase, medial region and trans phase. Cis phase, that is facing the nucleus, and trans phase is facing the cell membrane, and medial region is present between the cis phase and trans phase. Cis phase is the entry surface that is located nearest the nucleus, and it receives material from transport vesicles which are budded off from endoplasmic reticulum, and these transport vesicles, they contain proteins. Enzymes which are present here in the medial region, they are responsible to modify that proteins into glycoproteins, glycolipids and lipoprotein. Now, what are glycoproteins, glycolipids and lipoproteins? Glycoprotein is a protein with covalently attached carbohydrate. So, protein that is covalently bonded with carbohydrate, that is called glycoprotein. Glycolipid is a lipid with covalently attached carbohydrate, that is called glycolipid. And lipoprotein is a protein with covalently attached lipids, that is known as lipoprotein. So, this is the function of Golgi complex, that modify the protein into glycoprotein and lipoprotein, as well as it also synthesizes glycolipid. Trans phase, which is the third region of Golgi complex, that is called exit surface, and this exit surface is close to plasma membrane. It packages molecules in vesicles that transports out from the Golgi. In some animal cells, Golgi complex, that is present at one side of the nucleus, but in other animal cells and plant cells, Golgi complex, that is dispersed throughout the cytoplasm. Cells that secrete large amounts of glycoproteins, they have many Golgi stacks. Golgi complexes of plant cells, they produce extracellular polysaccharides, that is called cellulose, and cellulose is one of the component of cell wall. As we know that in plant cells, outermost layer is called cell wall, and cell wall is composed of cellulose, and cellulose is synthesized by Golgi complex in plant cells. So, this is the structure of primary cell wall, and primary cell wall is made up of cellulose microfibril. Inside the primary cell wall is here plasma membrane, and inside the plasma membrane is cytoplasm. So, primary cell wall that is composed of cellulose, and cellulose is synthesized by Golgi complex in plant cells. Next to primary cell wall is middle lamella. It is a cementing layer which holds all the cells together. In animal cells, Golgi complex manufactures lysosomes and glycoproteins. Glycoproteins that is secreted from the cell that is used to form extracellular matrix. In animal cells, Golgi complex secrete mucus that coats the epithelial cells. So, we know that epithelial cell, it is a single layer of cells, and this epithelial layer that has a thin film of mucus, and this mucus, it is secreted by the Golgi complex which are present here in epithelial cells. How the Golgi complex they work in animal cell and plant cell? As we know that Golgi complex, it has three areas, that is a cis phase, trans phase and medial region. Cis phase is facing the nucleus, trans phase is facing the cell membrane, and medial region is present between the cis phase and trans phase. Here is the medial region where the enzymes are present, and these enzymes are responsible for the processing of protein. So, secretory proteins that are assembled in the rough endoplasmic reticulum lumen are first transported to the cis phase of Golgi complex. These proteins then progress through the Golgi stack and undergoes successive modifications until they reach to the trans region, trans phase of the Golgi complex. They are packed into vesicles, and these vesicles are shipped to their destination membranes. What is the importance of carbohydrates in glycoprotein molecule? As we know that glycoprotein is a protein with a covalently attached carbohydrate. In some cases, carbohydrate component that may be a sorting signal, a cellular zip code that tags the protein and routing it to a specific organelle. Glycoproteins are packed in transport vesicles in the trans phase, and these vesicles they pinch off from the Golgi membrane and transport their contents to a specific destination. Now, what is the connection between transport vesicle and cell membrane? Vesicles they contain finished products like glycoproteins, glycolipids and lipoproteins, and these products they are packed in vesicles, and vesicles they transport these products for export from the cell. During this export, vesicle it fuses with the cell membrane, and the vesicle membrane becomes part of the plasma membrane, and glycoproteins are secreted from the cell. So, this is a vesicle that contains glycoproteins, glycolipids or lipoproteins, and when this vesicle it fuses with the plasma membrane, so the vesicle membrane that becomes part of the cell membrane, and glycoproteins they are exported, secreted from the cell, that is called exocytosis. Exocytosis is a process by which the contents of a cell are released to exterior through fusion of secretory vesicle membrane with the cell membrane. So, this is a secretory vesicle membrane. When the secretory vesicle membrane it combines with the cell membrane, now the membrane of secretory vesicle that becomes part of the cell membrane and the contents are secreted from the cell, that process is called exocytosis. Multiple paths of glycoproteins. Glycoproteins, that is the finished product produced in Golgi complex that are secreted from the cell during exocytosis, may be stored inside the cell and may be routed to various organelles. Which type of cell contains more Golgi bodies and why? Cells that engage in great deal of secretory activity, they have an abundance of Golgi complex. Examples are salivary gland cells, they secrete digestive enzymes, and immune system cells like white blood cells, they are involved in the production of antibodies. We have three pairs of salivary glands, that is parotid gland, submandibular gland and sublingual gland. In case of plants, Golgi complex, they have an additional role as compared to animal cell, and that role is they are involved in the synthesis of polysaccharides like cellulose, and that cellulose is incorporated in the cell wall. Let's watch this simulation to check into thoroughly the function of Golgi complex. What is the difference between Golgi complex and dictyosomes? As we know that Golgi bodies, that is a type of cell organelle, and it is composed of stacks of cisternae in eukaryote. So, in eukaryotic cell, these are stacks of cisternae that constitute an organelle which is called Golgi bodies or Golgi complex. Now, what are dictyosomes? Dictyosomes are individual cisternae which collectively form the Golgi bodies. So, dictyosomes are building blocks of Golgi bodies. In animal cell and plant cell, the distribution of Golgi complex is slightly different. Like in plant cells, dictyosomes are less tightly packed to form Golgi apparatus, they are dispersed in cytoplasm, but in animal cells, they are tightly packed together to form the Golgi apparatus. To sum up, Golgi complex modifies and packages proteins produced on rough endoplasmic reticulum. Structure and composition of Golgi complex. Golgi complex that consists of flattened membranous sacs which are called cisternae, and each cisterna has internal space that is called lumen. Golgi complex, it has three areas, that is cis phase, trans phase and medial region. Cis phase is facing the nucleus, trans phase is facing the cell membrane, and medial region is present between the cis phase and trans phase. Functions of Golgi complex. In plant cells, Golgi complex, they are involved in the synthesis of polysaccharides like cellulose, which is used in the synthesis of cell wall. Golgi complex, they are involved in modification of proteins into glycoproteins and lipoproteins. They are also involved in the synthesis of glycolipids. Glycoproteins, they are secreted from the cell, which is used to form extracellular matrix. Golgi complex in epithelial cells, they are involved in the secretion of mucus. What is sorting signal? Carbohydrate component may be a sorting signal, a cellular zip code that tags the protein and routing it to specific organelle. Amount of Golgi complex. Cells that are engaged in a great deal of secretory activity have an abundance of Golgi complex like salivary glands and white blood cells. Now, difference between Golgi complex and dictyosomes. Golgi complex, it is a cytoplasmic organelle that is present in eukaryotes, while dictyosomes are the building blocks of Golgi bodies. Let's watch this slideshow to differentiate between cis and trans phase.